Hi, on this video, I'm going to talk about the fourth and fifth standards of the science and engineering practices that we use in our biology grading rubric. And these are analyzing and interpreting biological data and using mathematical and computational thinking in biology. Now, a lot of times these will go hand in hand. There are situations where they don't, but usually when you're analyzing and interpreting data, you're going to have to do some sort of computational thinking at the same time. And that's why I'm talking about them in the same video. Now, when you analyze the data, you what you have to do is look at the numbers you collected and uh, do some processing with those numbers to understand what are some of the patterns or relationships that exist among the variables in the data. And if you do that well, while also explaining the significance of that, explaining what the data is saying, that is what the first criteria is talking about. The second criteria is relevant when you are analyzing data from like an experiment, when you have actual numbers. A lot of that raw data needs to be analyzed with computational methods, right? Mathematically, before you can see what the patterns actually are, which is where M5 comes in. And we'll talk about that in a second. So, when appropriate, you need to do that as well. So, it's almost like you can't really get M4 and meet the criteria for M4 for mastery without doing M5 if you're doing that in a situation where you collected raw data and have to analyze that data. And we'll talk about that in a second. But in other cases, what you might be doing instead is collecting data from multiple sources. And in those cases, you want to look at similarities and differences that exist in the types or what the information is saying from the variety of sources that you look at. And you're looking for consistency, right? Is it saying the same things when measurements or observations were taken? Now, you can also do that in an experiment by comparing different trials and seeing that is this consistent across trials. But you can also do this in textual analysis when you pull information from different sources and you say, are the sources saying the same thing? Um, the other thing that you want to do is not just stop there. So now I know I have analyzed the data. I know what the data says, but you also need to tell me what are some of the limits to your analysis. What are some of the things that you can't say because there's problems with how far you can take your data? Some errors in measurement, maybe limitations on your samples or the methods that you actually collected the data. What are some of those limitations? But it's not enough to just identify them to actually reach the benchmark for my biology class. You need to evaluate those limitations. So what that means, that word evaluate, means that you have to tell me why does this matter that there is this limitation, right? What are the consequences from my data that there are limitations? What can't I not say? What is affected by that? So instead of just saying, well, I only collected 3 samples, so that's a limitation, you can say, because I only collected 3 samples, I'm not sure my average is as accurate or consistent as it could be if I had had more data, right? So it could affect my averages, right? I could have had a limited number of observations, could limit how much I can generalize from this data, or I only work with girls and I can only make conclusions about girls in this lab because that's all the people that I looked at, right? And so that not just acknowledging the limitations of the data, but how those limitations impact your analysis. That is what evaluation actually means. And last but not least, make sure that you describe what matters about this data. What is the data telling you? What is the story that the data is explaining? What is it that you can imply from this data, right? So identify implications of the data or why the data matters. So analyzing data is about looking at your raw data, uh, doing some analysis or computation with that so that you can notice patterns or relationships between certain variables, and then identify in what ways the different trials or different sources of data are similar or different from each other. Explain what the limitations are and how those limitations impact your results, why they matter, and finally, what story is the data telling you? Why is it significant? So make sure you do each of those things in order to reach the mastery of that skill. Now, as I just mentioned, in order to actually reach this level, you might also need to do computation. especially if you are collecting a lab and you have this raw data. So M5, which is using mathematical and computational thinking in biology, there is two main ways in which you're going to use this in class. When you actually develop a mathematical model or you do computations in the context of biology, there's a few times where this happens when you're studying about microevolution, when you're studying genetics, when you're studying uh, taxonomy, you might have to end up doing some mathematical analysis. But it also happens every time you do a lab or experiment and you have to apply a mathematical routine in order to answer a specific question, in which in this case is what is the data telling me, right? So basic applications such as calculating mean, mode, variability, percentages, basic mathematical operations like that is enough to get the lowest level that I expect for mastery. But the highest level expects you to do more complicated statistical analysis, um, graphing to represent the data, or in the case of mathematical applications such as the Heidi-Weisenberg principle, the trying to predict the results of genetic crosses or calculate relationships in taxonomy analysis or things like that. That is examples of when you actually do math in the process of answering a scientific question. That is where you really want to take this standard. But in even in just normal labs where you just calculate descriptive statistics like mean, mode, standard deviation, percentages, then you're also doing this skill. And that's what you want to do when you want to do an experiment. Now, at the higher levels, you can also do some statistical analysis for significance or for correlations. That would actually show you if your data is what is the data is actually is meaning and it's actually relevant. Can we actually trust this data? That would be an example of reaching the higher levels with experimental data. But you're going to have to do at least the basics in order to get M4, which is why I mentioned in the same video as this one. Um, there are lots of resources that I have available that talk about how to do this in your lab report resources. Um, you can see how I did this if you look at the lab report samples. And if you look at the lab report videos or writing videos, I talk about how the data analysis section actually work on that. And in the lab report explanation, it talks about how in the sample you can see me doing it. And also there's the guidelines that gives you criteria for what that you should hit. So these are some good resources. I also have a document that's linked in the specific resources that talks about how to represent physical phenomena well. And in here, it gives you some examples of some things you might want to do in order to represent your data, and you can look at that. You can also look at these examples of graphs, different kinds of graphs. We have line graphs and circle graphs and pie charts. And I also have a separate playlist of videos that talk about how to analyze data in more detail. So you can look at any of these resources if you need further support with this skill. And I recommend that you do in order to do that. And don't forget, there's a complete separate podcast series that talks about each step um, of the process of writing a lab report, and including analyzing the data. And there's also a lab report sample where you can see me doing analysis of data for other experiments. And there's two samples like that. One of them is in physics, the other one's in biology. And you can see how data analysis looks like by looking at those samples. So if you need more help, make sure you reach out to me. I'm always available. And thank you so much. Good luck. Don't do anything that wouldn't make your mama proud.